Okay.
All right, yeah, and now the program works. Right.
Okay. Awesome. Sorry, let me get to the right screen.
So share my screen. Okay, making recordings in progress. Back to here. Give you control.
Okay, I have it. Okay, everyone, we're gonna get started. So we're just gonna go through everything that's gonna be on the test. We'll start with the sensory neuroscience part. Okay, so all sensory processing begins in receptor cells. And what happens with any kind of sensory receptor is that it's going to depolarize a little from every kind of stimulus that it receives from the environment. So all these mini little depolarizations are called graded potentials. And if the graded potentials add up to a potential that's big enough to generate an action potential, then it'll actually generate an action potential. And
I took back control for a second. Sorry. Give me 1 second.
oh, you're good. No worries. And the threshold, the threshold at which like it becomes from a grade potential to an action potential is called the threshold potential. So everything before the threshold is called a generator potential or a graded potential. Everything after the threshold potential is an actual action potential that sends signals to the brain, and that's something that you actually perceive. Okay.
Okay. You should have
Could
control.
you just go to the next slide? Okay. Do I have control?
Yeah. Now you should.
Okay, cool. Okay, so the intensity of a stimulus is communicated to the brain in different ways. So you can change the frequency of the stimulus. So if you have a lot of action potentials that happen one after the other, it tells the brain that the stimulus is more intense than a regular one. You can have multiple different neurons send action potentials. So if you're having a lot of different action potentials happen at the same time from different neurons, the brain's going to understand that it's a bigger stimulus than something that might have been smaller. And also, the brain has different types of neurons that respond to different ranges. For example, with touch, you have different touch receptors that respond to different kind of intensities, or different pressures. Some respond to deep pressure and some respond to really light pressure. So depending on which receptor is activated, that's another way the brain can tell what kind of stimulus is being given. So these are the 5 touch receptors that you have in your skin that the brain uses to detect touch. So Meissner's corpuscles, which are going to be these ones over here, detect light touch. The Merkel discs, which are over here, respond to like edges and isolated points. And what we mean by that is like textures, kind of like if you ran your hand against something super sharp, detecting something like that, versus Meissner's corpuscles would be like the actual pressure on your skin. The Ruffini corpuscles detect stretching of skin, free nerve endings respond to pain, heat and cold, the ones over here. And the Pacinian corpuscle is the one all the way down here. That's the one that responds to vibration and deep pressure. So with the Pacinian corpuscles, it kind of works like this. When the skin is vibrated and stretched to the extent that the Pacinian corpuscle is detected, which means it had to have been like a pretty deep vibration, the channels themselves are stretched and sodium ions are like allowed to go through when they are stretched, and that's how it gets depolarized. Okay. So with touch systems, you have tonic receptors and phasic receptors for every sensation. So tonic receptors are receptors that will fire for as long as you are touching something. So this kind of pattern is an example of a tonic receptor. So an example of that is that if I was touching my laptop, all the tonic receptors in my hand would continue to fire active potentials for as long as I'm touching the laptop. And that's how my brain knows that I'm touching it. Phasic receptors are receptors that respond to changes. So with the laptop example, if I was touching my laptop, then the phasic receptors would fire really quickly in the beginning to let my brain know that something has changed, and then they would stop firing after that. Okay. Each receptor has a receptive field, so an area of the skin or an area of the body that that receptor is going to respond to. And it's for each sensory neuron. And these will be different depending on what type of sensory neuron it is. But in general, there is a receptive field in the middle, like the light green, that will fire when there's some kind of stimulus in that area. And the area directly around it is actually going to decrease its firing so that the contrast between the two signals can be kind of increased. You're going to increase the contrast so the brain can more accurately feel whatever is happening or detect whatever stimulus has occurred in the environment. So there's two different tactile systems that we see in our skin. The first one is called the dorsal column medial lemniscus pathway. And this pathway has neurons that detect touch, vibration, and proprioception. Touch and vibration are touch and vibration. Proprioception is your own kind of awareness of where your body is and how it is oriented. And the sphenothalamus pathway, on the other hand, contains neurons that transduce pain and temperature information. So it's really important to understand exactly how these pathways move through your spinal cord and into your brain, because you realize that if you notice, they cross over at different parts of your brain. And this becomes important when we talk about different tumors and lesions that can happen along this pathway and which side of the body it's going to affect. So with the dorsal column medial lemniscus pathway, all your sensory neurons will enter the spinal cord over here, and then they're going to proceed up ipsilaterally on the same, oh no. Okay. Then they're going to proceed ipsilaterally on the same side until they hit the medulla in the brain. Once they get to the medulla, they're going to cross over to the other side. So they're now contralateral. And then they're going to move up to the thalamus into the cerebral cortex. And that's where this happens. So just remember that it crosses over at the medulla over here. And with the sphenothalamus pathway, the the neurons are going to enter the spinal cord, and then immediately at the spinal cord level are going to cross over and become contralateral within the spinal cord, and then move up ipsilaterally on the same side through the medulla, through the thalamus, and up to the cerebral cortex. So just remember that the DCML crosses over in the medulla, and the spinothalamic pathway crosses over at the spinal cord level. Okay. So touch information is conveyed through the spinal cord, and if it's in your face, they're going to be picked up by cranial nerves, and this information is going to consolidate in the brain, in the spinal cord, up to the brain stem, move through the thalamus, and move up to the somatosensory cortex. And that information is going to be moved to different sensory cortical areas depending on what kind of information it is and what brain regions need to process it. But the major pathway is it moves through the brainstem, through the thalamus, and to the somatosensory cortex. The somatosensory cortex itself is broken up into different areas. Different areas of the cortex represent different parts of your body, or neurons from different parts of your body. Areas of your body that have more neurons than other areas are going to have a bigger allocation of space in the somatosensory cortex. For example, the area for your hand is much bigger than the area for your forearm because you have a lot more neurons in your hand, or sensory neurons in your hand, than in your forearm. And that's just kind of how it works. The cortex is subject to change depending on like experience and what happens. For example, in here, someone's hand was amputated and their somatosensory cortex kind of adjusted for it. See here.
Do take note on where the face is in relation to where the arms are and the feet are. So know that like the tip top of the head has its feet, and then it falls down where the face is on the sides. But you don't really need to know how large those individual places are. Just know, where the feet and then where the head is, just so that you know in relation to where things are.
Okay, let's talk about pain. So there's two kind of different types of neurons that transduce pain. You have neurons that are called A delta fibers and neurons with C fibers. The difference between these two is one of them is myelinated and the other one is not. The A delta fibers are like the free nerve ending. These are neurons with free nerve endings that pick up the pain. And because they're myelinated, they transmit that information really quickly to the brain. So they detect sharp pains versus C fibers that are unmyelinated. So they will detect dull pain and they're going to transmit this information pretty slowly compared to the A delta fibers. Okay. Capsaicin is a material that's found in chilies or in other really spicy foods that you eat. It's a special chemical that's picked up by TRPV1 receptors. And it's another example of pain perception because capsaicin are picked up by pain receptors in your brain or in your skin. Oh, sorry. In your taste receptors. Hold on. Okay. So free nerve endings in your skin pick up pain from injury. When Arthur,
Yeah,
could you help me out here?
sure. So, for just to see like what's important in this slide.
Yeah.
Okay. Um, so we never really talked about the mast cell, so that's really not too important. And when it comes to trauma, we also don't really talk too much about the prostaglandins or the leukotrienes. So really, the important thing is substance P. So when we get pain, substance P is released into the blood, and action potential is also brought back to the brain. Substance P is said to be a neurotransmitter, but it really only is released when pain, through chemicals, tissue injury, or temperature burns. It runs through our C fibers, and really the only important part is the fact that we have our fibers that sense pain and they release substance P. Histamines, maybe, so that we can prevent swelling and also give information about that. But essentially, this slide really just is talking about substance P, because we'll later talk about the integration and the canceling out of the sensory of pain.
Okay, there's different areas. Hello? Okay. There's different areas of the brain that can mediate our perception of pain. So your typical pain pathway is the one we talked about before, the sphenoidothalamic pathway that moves up through the thalamus and into the somatosensory cortex and other areas of the brain that process pain. At the same time, there are areas of the brain, like periaqueductal gray, that can modify the ultimate pain perception. For example, the periaqueductal gray has like opioid receptors near it. So the integration of both what you feel through the sensory pathway and the modification through other areas of the brain are what ultimately increase. So these two pathways kind of modify the ultimate perception of what level of pain you're in.
Yeah, and the really important part is the periaqueductal gray, which will be the area that is going to be sending information to cancel the sensory of pain.
This is kind of what we talked about before. The sphenoidothalamic tract will transmit pain information about pain to the brain up to the somatosensory cortex. And the cingulate cortex and the periaqueductal gray are areas of the brain that modify this perception of it, that modify pain perception.
We got a question on what does the RVM stand for in the last slide. And I actually don't know, do you?
It stands for rostroventral, I believe rostroventral medulla.
Okay.
I will check on that.
But yeah, it's definitely not an area that she's going to test on. The periaqueductal gray, as it shows up in this slide and it's highlighted, that's really the important takeaway from that last slide.
Okay. So pain is processed kind of at different levels. We talked before about how there's different areas of the brain that can reduce our perception of it. This is kind of like an example that takes you through how pain is processed. For example, if someone broke their wrist, the physical trauma that comes with breaking your wrist is what the spinothalamic pathway is going to send to the brain and say, hey, like we're in pain because our wrist is broken. However, there's, depending on your situation, or the situation that you're in, your brain will modify how much pain you feel. For example, if you're in the middle of a basketball game and you know it's super important, then you're not going to be likely to spend too much. You're not going to focus more on your pain. You're probably going to be focused more on the game. And whatever your cognitive biases are towards your situation, if the person who's playing thinks that men don't cry, then they're also going to be less likely to focus on their pain or less likely to concentrate on it. So that's also going to affect ultimately how much pain the person is in and what they do.
It's also the end point is the motor response mechanism. So it's our response to that initial pain. So we're feeling that we got a broken wrist. So the men don't cry side of the cognitive system is just that we might still feel it, but we're not going to respond on it. Mostly the motivational affective system, which is what's coming back towards the central of the spinal cord processing, is the fact that our body realizes that it's such an important game. Or say that someone was running for their life, they break their, or not break their ankle, but like sprain their ankle, and they still have to run because their life's dependent on it. So they're going to be pumping adrenaline, pumping all sorts of chemicals, so that we will ignore the feeling of that pain so that we can continue to run. So that's like that motivation to stay alive or to like win that championship game. That'll cancel our feeling, but the cognitive system is really just stopping the response, the mechanism. So our motor response to cry or to like kind of like grab our leg and our wrist and just feel hurt and want to stop doing something. That's more of like the thinking side of it, but the chemical side is going to be that motivation.
Okay, so the gate control theory of pain kind of talks about how you can modify a painful stimulus by activating other stimuli around the area. So if you have a painful stimulus in one area that's activating C fibers on pain neurons. If you also have an A beta fiber, which is like a normal touch fiber, if you activate those neurons, these neurons will ultimately inhibit the interneurons that are processing it. So if you have another stimulus that adds to the painful stimulus, then it'll reduce the total amount of pain that is perceived. Some other factors that influence this are your attention and emotional cognitive factors that we were talking about before. Those will also inhibit or modify how much pain that you ultimately perceive. So transcutaneous electrical nerve stimulation kind of plays on this idea. When people have pain in an area and you also use this transcutaneous electrical nerve stimulation in the same area of the pain, and you stimulate the touch neurons instead of the pain neurons, then you're going to ultimately inhibit you're going to introduce like a counteractive system that ultimately reduces how much pain that you perceive. So things like meditation, acupuncture, massage, and physical activity will all play into the gate theory idea and reduce the amount of pain that you feel. So pain is important because it tells us when something is wrong and when we need to change our body position or move our muscles. And if our pain perception is compromised, then we get things like, you can get things like gangrene, and due to us not perceiving when we need to be moving our hands away or something to that nature. For example, when people have congenital insensitivity to pain, they won't move their fingers away when they're in danger, or their muscles will atrophy. And that's why it's important that we have our sense of pain intact. Congenital hypersensitivity for pain is also a different variation of this. When your pain receptors are too active and they're always firing, it's really difficult to function in daily life because you don't know what is dangerous and what is not dangerous. These mutations arise from the sodium 1.7 channels. These are the channels that are responsive to pain. So chronic pain is when pain persists for longer than it should, long after an injury has healed. There's different kinds of chronic pain. Neurogenic pain is pain that results from damage to nerves. So damaged nociceptors are going to continue to send signals to your brain that tells it it's in pain, even though the injury is healed and there's no reason to anymore. Psychogenic pain is not pain that arises from the neurons themselves, but other places in the pathway. And it's going to tell your brain that you are in pain even when you're not. Reflex sympathetic dystrophy is a syndrome that occurs when this process of having pain right after an injury and having it disappear once it's healed is disrupted. So stage 1 of this disease would have pain that lasts a few weeks after an injury. So it would last for a little longer than it really should, and then it would subside. At stage 2, the pain becomes severe and more diffuse, and you're unable to really feel, you're unable to turn off that pain response and that inflammation response, which is going to damage your muscles and your hair and that entire area. And just as a review, the DCML pathway is the one that's responsible for touch, vibration, and proprioception. It crosses over in the medulla. And the sphenothalamus pathway will cross over right in your spinal cord, and it's responsible for pain and temperature.
All right. Do we have any questions on sensory before we move on to the eyes? Okay. All right. So the eyes and vision. Really important part is the retina, is where we're going to hold all and contain all of the photoreceptors, where we convert light energy, and that changes into the membrane potential, and that is going to be what gives us the information sent to our brain. to understand that light. Definitely know that a very big important part of the transmission of information to the brain, occipital lobe, is that it passes through the lateral geniculate nucleus and the thalamus, and that is very important as a stopway point when you are organizing information to send it to the primary visual cortex. So we first start off with the eye itself. The pupil and the iris is going to be, what we normally see with the eye, the color, darkness. Pupil widens when we are in low light. It constricts and gets tighter when there's tons of light, and that's going to, give us brighter or less bright images based on our surroundings. The iris is the color, which is basically a muscle that does that dilation or constriction to allow more or less light in. Cornea being this area that's going to hold the lens that is going to refract the light properly to hit and get into the retina, which is the back of the eye. That's going to be processing all that information. So this allows us to see far away, close by. The conjunctiva is this area here which prevents things from kind of rolling to the back of your eyes, and if you get some stuck in your eye like contacts, it won't go to the back of your head because of this membrane, but it's not too important. I'm pretty sure she wouldn't really necessarily test on that. We do have muscles for our eyes, and something very important for that relay of information is the optic nerve. Straight from the retina, we're going to send our information to the nerve, and we'll get more in the information processing in just a moment. So again, the important part here is the iris, the lens, cornea, knowing that the lens is in the cornea, and knowing the retina, fovea, and optic nerve. Sclera is that white outer side, but we shouldn't really talk about that too much, or any of the liquids, or too many of the muscles in there. After that, information comes in through the light, and we sense it in our retina cells. The information is going to be taken down through the optic nerve, which is going to cross and indexate partially in the optic chiasm, and then it's going to turn into the optic tract, goes into the LGN and the occipital lobe. But first, let's continue in the retina. The light actually comes in. There is some matter in the way, but the light makes its way through, and the rods are actually at the end, which pretty much is at the back of the eye. We have these two other sets of cells that are in the way, but especially in certain areas, they do get pushed away so that we get really clear view of the light. But here's the order. The light hits the rods and the cones. They will process that information. It'll be given to the bipolar cell layer, then go to the ganglion layer, and the ganglion is going to take it out through the optic disc and take that information to make the optic tract, or the nerve and the tract. So know that the receptors are going to be what's going to be converting the light into energy into a membrane potential. And the ganglion cells are what's going to be firing action potentials, and that's going to be relayed to the brain. So ganglion cells with action potentials will respond to the light. Receptors or convey light energy into a membrane potential. So the membrane potential will be understood by the bipolar cell. That's going to be activated or inactivated to the ganglion cells, ganglions having the full action potential as we know it. Bipolar cells are just kind of that in-between cell. So you definitely need to know the order of their photoreceptor, bipolar, ganglion. the horizontal is going to kind of just modify that information so we can get a finer detail on certain things. We can understand edges of things and see, more of a defined understanding of this information. She doesn't test so much on those two, but do try to remember that the horizontal cells is in that synaptic area the um, of the photoreceptors and the bipolar cells, and the amacrine cells are bipolar and ganglion. I have a question that says, so does light go all the way to back to the rods, then moves backwards? So yes, the light essentially will come through all of these things, kind of hit the back of that eye, or like the matter in the eye, and then that information will be processed here. The light will be turned into a membrane potential. The membrane potential will be processed by the bipolar cells. It will give action potentials to the ganglion cells, and those action potentials will be sent to the brain. So the light comes through all of this, it's understood here at the rods and the cones, and then does come back up, and that information will come over here and go sideways to make its way over to the optic disc. So we have a lot more rods than we do cones, but cones will be our very fine detail. Rods will just be our light and our dark, and we won't get very great resolution from that. Especially like if you imagine when it's really late at night and you are trying to see in the dark, you can kind of see when it gets really dark, but you really can't make out a lot of detail. You can kind of just tell that maybe there's something there. Maybe you can see some darker shadows within the darkness, but you don't really know necessarily what it is. But when it comes to your cones, when you can see a lot of difference in color, you can see a lot of difference in edges. Say there is tons of light and it's kind of just really, really, really bright. You're still able to make that differentiation between color. You can see the outlines, you can see kind of some more detail even if you have an excess of light. But when it comes down to having barely any light, your cones really can't do too much. But when we have high spatial resolution, because when there's a good amount of light, you can make out tons of detail. We're able to read, we're able to make sense of images because our cones are actually are somewhat insensitive, and allows us to kind of process and make definitive clarity. Do notice that the rod is a lot larger than the cone. That is because cone is sensing individual colors, and the wavelength is it varies a lot more, and the rods kind of don't discriminate. They see all of the wavelengths of light, and so the entire spectrum is there, and also it allows it to be very sensitive to light, because if light hits it a little bit, all of this is going to activate, meaning we're going to have a strong membrane potential, as opposed to the cone. Even with a little bit of light, it might not be enough when all of this is activated to really make a strong membrane potential. So the fovea is where we see the best, which means we have high visual acuity, or the best visual acuity. This is the area, if you see down here, we actually get the ganglion and bipolar cells pushing themselves out, where the light that's coming down is being able to go to the rods and the cones straight, as a straight shot. So these cones mostly will get no interference. The light won't have to go through those ganglion cells, so we get a very clear image in our focal view. That's why our peripheral view is kind of not as fine detail as when we're directly looking at something. So that's where our fovea is where these ganglions push themselves away. And if you notice here on this slide, it is showing that cones are mostly in this area. We're mostly getting a lot of color, a lot of fine detail, as opposed to our peripheral, where we're not really able to see so much fine detail, and it's mostly just vagueness, mostly vague shapes. We just know that something's there, but if you try to look at something with the side of your vision, you're not really going to be able to read or really see too much. So light transduction is going to be using G protein, so it's a cascading effect. It's going to activate something, and that activation, the light activating the photopigments, is going to break down GTP, and that is going to cause a large effect, as opposed to if it was an ion channel where it wouldn't magnify itself, it would just stimulate the one channel, and that one channel depolarizes, as opposed to having a cascading effect where one change activates one enzyme that activates tons of molecules. So I think there's a little more detail in the next slide about that. So opsin, or it could be rhodopsin or retinal, is in this cis configuration, and when light hits it, it turns into a trans configuration when it's activated. And this activated state is going to activate our G protein transducin, and that is going to produce an enzyme that is going to break down the cyclic GMP into regular GMP, and that's going to cause our sodium channels to close. So make a note that even in darkness, our receptor cells are constantly activated. The sodium is constantly poured in, which is something that's different than how we understand most cells. Normally, our cells are closed until we get activation, or our channels are normally closed until we get activation. In this situation, the channels are constantly open, and once we get activation from the light, we're actually going to close these channels. So make sure to understand this process, that light comes in, rhodopsin changes its configuration. That's going to activate an enzyme called transducin. It's going to lead to the breakdown of cyclic GMP into GMP, and that's going to close our sodium channels, because the thing that is holding our sodium channels open is cyclic GMP. So the moment that you break down the thing that is holding the channel open, you're going to have a closed channel. So in the dark, we are getting constant membrane, positive membrane potential. So we're in a constant depolarized state. When we get light, we're going to then hyperpolarize, because we are removing the availability for sodium to come into the cell. When sodium is coming in, we are depolarized, and then once we break down that cyclic GMP, it's closing our sodium channels, and that is going to lead to a hyperpolarization. So we're going to stop our membrane potential, and it's going to be negative again. So here's a rundown of how it happens. is phototransduction. Light comes in, the receptor protein rhodopsin, which either opsin or retinal, cones and rods have different names, gets activated. That activates the G protein transducin. It's going to create an enzyme that is name is a little unnecessary, but if you need to know it, it's the phosphodiesterase. It's going to decrease or start to break down cyclic GMP, and that's going to lead to the closing of sodium channels, and the membrane hyperpolarizes. So that is the cellular side. This will go down. We'll start with the that information gets then all right, here. The hyperpolarization of the photoreceptor is going to stop activating the bipolar cells. The bipolar cells will understand that it's no longer being activated by the photoreceptor and will tell the ganglion cell that it is receiving light. The ganglion cell will then bring information, and it'll do so in this way. One eye is going to be able to see both hemisphere. Basically, your vision is able to see about 30% of the other eye's information. We're only blocked by our nose, or else we'd actually be able to see quite a lot more. I'll draw a diagram on that because it is a little important to understand what part of the eye sees what, how much of that information gets brought back. As you can see, we have a visual field, and our occipital lobe on one side is going to see the opposite side's visual field. However, you notice that both eyes are able to see that visual field, and we have an area called optic chiasm that is going to give part of that information to the other side of the brain, but it's still going to keep its certain side. No, I can't zoom in. All right, I'll draw it out. Let's see. Can I annotate? Okay. So we will have a visual field here and a visual field over here. One eye is going to be able to see about this much of one, while the other eye is able to see about this much of the other. So we are getting information from this side, and this side of the eye is going to get information from that side. So knowing that half of your eye is going to be receiving information from one visual field, and the other half of the eye is going to be getting information from a different visual field. So here, in this area that was crossing, is going to be the optic chiasm, where information from the nasal side, so know that the nose, that's an awful drawing of a nose, but imagine that's the nose. So this is the temporal side of the retina, and this is the nasal side of the retina. And this is going to be our right visual field and our left visual field. So know that the opposite retinal side is going to see the other visual field. And so the nasal retinal is going to see the right visual field on the right eye, and it would be the opposite for the other eye. So the nasal side of the retina is going to see the same side's eye's field, while the temporal side is going to see the other side's visual field. And so that temporal side is going to see opposite visual field, and so that visual field is going to go to the same side of the eye, while the nasal side is seeing the same eye's visual field, and that same side is going to be given over to the other side of the brain, because the left side of the brain is going to see the right visual field, and then the right side of the brain is going to be seeing the left visual field. So attempt to be able to draw out a diagram like this for yourself, where you kind of just make something that you can understand, that you can see that when you have an eye and you can draw a pupil, and when you're seeing the back of the eye, you can draw a straight line to the direction it is viewing. So when, say, we're looking at an image of something, say we're talking about an arrow, the one side of the light that is coming from the right is going to come into the left retinal view. So we're going to get an opposite view in the eye. So know that whatever is on one side is going to be on the opposite side of the eye, and then that is either going to go to the same side of the brain or the opposite side of the brain. If you can remember that the nasal side is going to be the one that switches and decussates, then you'll be able to draw this pretty easily. So knowing that the eye sees the opposite visual field than the retinal side, and knowing that whichever eye it is, wherever the nose is going to cross, and whatever side the your temporal or your ears are is going to go straight to the same side of the brain. So knowing that same side goes same side of the brain, we can draw our eye, and that is going to be the opposite visual field. Does this make sense? I hope it kind of did. Please ask a few questions if you have it. Maybe I can clarify, or maybe Divya can give it a try. But it is likely that there will be a question on this, because she does like to test on that.
No questions, guys? I didn't realize my video was on.
Okay. I don't know how to remove all that I drew now, other than going individually. Oh, okay. Wow. Okay. So when we were talking about the decussation, and we're going to bring one side over to the other, and we're bringing the information to from the same side or crossing it over. this is each going to come to the LGN. And we're going to have that LGN on both sides of the brain. And that is going to be around this area, around, the central side of the brain after we have our optic chiasm. And the LGN, which is the lateral geniculate nucleus, is going to take that information from the opposite visual field and is going to relay it to the to the occipital lobe. Divya, do you know if they need to know anything about the layers, or just magno and parvo?
You just need to understand that there's a magnocellular and parvocellular.
Okay. Is there a slide? There's not a slide on that. Okay. So important side is this right here. The optic radiation is basically the layering that is going to occur with our magnocellular and our parvocellular, and it doesn't really tell you exactly what those are, but magnocellular is going to be motion, fine detail, able to track things, while the parvocellular is going to be some color, a little bit of shape, and just less important detail. So this last slide is a better representation where we're getting information from one eye, and it's showing how one eye gives information to both sides of the brain. But it's because of that visual field coming from one side and the other side that this outer side is going to come and stay on the same, and that nasal side is going to, like I say, come over to the other side. Okay. That's motor. Divya, do you want me to continue with the motor, or do you want to take over for a little bit?
Yeah, you can continue.
Okay. There's still no response from Mustafa. Yeah. Does anyone have any questions on the visual? Did I do the explanation justice? Do we want any more elaboration on any specific side of it? Because I know that specifically the cellular stuff can be quite a bit. It's a little different than what we're used to with action potentials and how we understand the cell to be activated and deactivated. So that is definitely an important side to know. And as well, the visual hemifields or visual hemispheres coming to the back of the brain, knowing how that crosses or doesn't cross is very important as well. Okay, so Cindy asked about the importance of the magno and parvo. So magno is this bottom side here. It's the darker cells. They're big. They're really actually important. They're going to be what we see for motion. So it's kind of that animalistic important side of it, where we need to see when things are moving. Is something going to attack us? Is something going to, or is something that we're hunting? Are we able to see it? So we're able to tell if something's moving. Is something being thrown at you? Can you notice that and catch it? It is that fine edged detail. So if you're able to see something moving and changing shape, you're able to lock your eyes on it and kind of make sense of it. And then the parvocellular is just going to be color. Really, color doesn't matter so much when something is going to come at you, but color it is is kind of a secondary information. It doesn't seem that she really cares for you to know big, deep detail on that, since she didn't really give you a definition of what those cells are and they're different, what differentiates them. The extent, I guess, would be this slide, just knowing that there are 6 layers. Maybe knowing that how they're numbered, so they're numbered bottom to top. Those magnocellulars are going to be those 2 layers on the bottom because they're bigger, and then parvo being the 4 layers on top and they're a little smaller. And both sides of your brain has it. So it's your thalamus, knowing that they're in the thalamus, and this is how it's going to split that information and send it to your occipital lobe in the back of your brain. So it's basically right. It's right here. Your lateral geniculate nucleus. You see how there's multiple different axons. I mean, it's simplified down here because it's just a small image, but it's going to split into different portions in the lateral geniculate nucleus. It's going to layer it in 6 different layers, and those layers are going to be given in different areas of the occipital lobe and how we're going to process that information. Okay. So with our motor system, basically we have motion that we want to do. Actually, give me one quick second. Let me just reset the recording.